Yeah, so I'm hoping to share with you a little bit about what I mean by the outsized is just their large role that often is overlooked sometimes in aquatic ecosystems. So as was already stated, I work in the field of microbial and viral ecology, and we address questions under this giant umbrella using three main categories of experimentation and different methods. And so largely we use what's called omics. And so omics, when we just say omics instead of making it more specific, it's really the collective technologies, right, used to understand what's going on at either the cellular level, population, community level. We also do more traditional cultivation-based methods, and this is to mostly pull in new model systems into the lab. And I say new, maybe emerging model systems might be the more appropriate term. We hope they're emerging and that more people will use them. But the real reason why we're doing this is because oftentimes the model systems that are in labs are not those that we can readily go back and sample the environment. And since a lot of our work actually is environmental, we still do a lot of field-based research to identify kind of the fundamentals around viruses in different ecosystems. We like to have those systems where we can constantly go back and reevaluate and make sure that what we're finding in the lab is still what we're finding in the environment. So it's a little tricky, but we hope that we have some really good candidates, and I'll show you a little bit of data on one of those today. And then we also do quite a bit of work of either utilizing, modifying, or developing what are called environmental assays. And so this is to hopefully generate numbers, rates, things, rate measurements that maybe mathematical modelers and others could use in their models, because often, particularly for viruses, there's stagnant numbers used, some of which come from studies in E. coli, which have nothing to do or any relevance with the marine environment. And so what we're hoping to do is start to build these numbers and go back over and over again to different environments and to try to understand just some very basic fundamentals in terms of how many viruses there are, who they're infecting, and what does that mortality mean to the greater community that's present. And so I know that you guys already heard from Forrest, so I just have one slide of introduction on viruses, just to kind of remind you, and then I guess I do have a couple peppered throughout the talk too. But what do we know about marine viruses? So they're very abundant. They're typically in any environment within the marine system. So if you're looking at surface on down, they're typically at least an order of magnitude greater than a cellular component, which often the next one is bacteria, right? Generally, you'll see a lot of times people say there are about 10 million per mil of viruses, and this is really taken from surface productive areas, but I like to use more that there's usually at least an order of magnitude greater viruses present. Viruses, very general, cannot reproduce alone, right? So they require some form of the host machinery in order to replicate their genome, make their capsids, which is what they usually house themselves in, their genomic material in, and complete their life cycle. And so they have, oh, of course, there's, gotta get used to the skin, it's been a while. The pictures over the thing, so hopefully, can you guys see my arrow, I guess? Yes, okay. So there are three main lifestyles or modes of replication. And so there is the temperate and prophage, which is where this typically happens with bacteria, but it can also happen in other systems. And this is where the genome of the virus is either incorporated into the host, or sometimes it's even kept as an episome, kind of like a plasmid or just another piece of genomic material within the cell. And it can stay within the cell for multiple rounds of cellular division and replication before something typically induces it to pop out and to start making its proteins and making its progeny. There's also the more traditional, which is the lytic replication strategy, which is where the virus injects its material or it gets brought into the cell depending on which virus it is. And it immediately starts to undergo replication. It builds itself back up, it's called packaging, until it reaches a certain number typically, and then it bursts the cell and releases its progeny back into the environment, right? And there's also a third one, which is gaining a little more traction in the marine system as we get a little better, I think, with our methods of how to look at this, because it's very difficult to track persistent infection. And so persistent or chronic infection is where a virus, it's sort of similar to prophage, right? Because they can infect and they can hang out there. So I don't know how old you all are, but we all got chickenpox when we were little, and chickenpox is one of those viruses that will stay with you forever. So it is a chronic, persistent viral infection. So it's one example, and it can come out later in life as, say, shingles often, which is a different kind of infection. But you have it forever, typically, once you have this kind of virus. And so we're learning, and I'm not going to talk too much about it today, but we seem to be learning that this might be a strategy for diatoms and their viruses, at least the ones that we are studying in the lab. So in their infection, right, they are clearly important agents of microbial mortality, and this can have big impacts on the diversity of the system, right? So if they're infecting certain populations, that can change the community as a whole or the food web as a whole. In doing so, they also can influence biogeochemical cycling, right? So the cycling of major nutrients, minor, but thinking of carbon and nitrogen. They are very similar to cells in that they contain protein, DNA, and RNA. They are different than cellular organisms in that you and I and every living thing that I know of has a double-stranded DNA genome, right? And viruses, though, are super unique and amazing, I think, in that they can have all these different kinds of genomic materials, all of these up here. So they can be DNA, they can be RNA, and they can also be double-stranded versus single-stranded. And so they go through, they really just harnessed, right, the genetic ability of replication, I think, which is kind of cool in using all of these. And I'm going to share with you a little bit about some of what I like to call other viral categories that are often also not discussed, because the majority of viruses that we have the most data on, particularly in the marine environment, is double-stranded DNA. But I'm going to talk to you about some positive sense single-stranded RNA viruses a little later. Through their infection, they can also go through a process called transduction, and that process can lead to horizontal gene transfer. So this happens sometimes when they're repackaging their genetic material. They can bring in some of the host genetic material into their capsids, and then they can add that back to the population, right, of their hosts, and they can transfer genetic material that way. This process is also one that's been utilized in biotechnology. So they now have viruses that they have manipulated in a certain way, to transfer genetic material without killing the cell. So this is commonly used in lots of different labs to introduce genetic material into new cells. And then finally, kind of along those lines, is a process called Lysogenic Conversion, which is the way that viruses can confer a phenotypic adaptation. So one example of this is with Vibrio cholera. So there's a toxin, right, that can make you very violently ill, but that toxin is actually carried by the phage. And so if you have a Vibrio cholera infection, but no phage and no production of those proteins, that toxin, then you're fine and you won't get as sick. But if it does have that viral infection, then those proteins, right, are being, and those genes are being transcribed along with the host. Oh no, what did I do? Oh, yes. Okay, so you guys have, I'm sure, seen this in many different fashions already. This is just one because I like to show it because it just shows a lot of different connections. So, right, you guys have heard of food chains, which is describing the energy transfer, energy flow within a system. And so you start at the base of the food chain in the marine system, that would be phytoplankton. And then you go to bigger organisms like grazers, fish, bigger fish, and on up. But really, it's much more of a web somewhat depicted here, where you have all of these different connections, and viruses fit in all of these different connections, which I think is also pretty interesting. So there's viruses that infect bacteria, plankton, zooplankton, microzooplankton, and even the fish and on up. And so one area that we like to try to understand more, right, through our assays and our omic studies is to say, well, what is that role out there in the environment, and do they play a big role, or maybe they don't? And, you know, or maybe it's a synergy between both the nutrient flow, maybe that phytoplankton are seeing, as well as infection that are causing sort of their dynamics to change over time. So a little bit simpler view, again, is something here, you know, I think I meant to take this one out, sorry, this morning, but it's sort of showing it again in a more simplified version, and kind of, at least this is sort of the one that I always go back to when I start to think about what virus role might play. And I do this because largely we're looking at viruses of either phytoplankton or bacteria, and we look at them primarily in regions of productivity. So our phytoplankton virus work is largely done in upwelling environments, or at least in the surface ocean, right, where phytoplankton like to be and be productive. And then we look at bacteria and viral infections in the deep ocean. So down at hydrothermal vent regions where you have chemistry, right, causing the primary producers to sort of produce all the different nutrients for the system. And so this is just a way of looking at all those different connections and how we can kind of think about those a little bit more. So ecology, and what do I mean by that? I think you guys have probably already either heard or probably had whole lessons on ecology, but just in case it's not as familiar, just basically, right, it's the study of relationships between organisms, or in my case, typically thinking about viruses and their environment. And so I put up 3 different questions, which I hope to touch on a little bit throughout the next few slides. So if we think about, right, the biological organization here, where you start at the very smallest from atoms and you work yourself up to maybe cells. And then if you're multicellular organisms, you have things like tissues and organs, then organisms, populations, and communities, right? And so on a community level, or a community ecology level, this could be the study of viruses in the ocean. Basic. Also very fundamental is how do they change through space and time. If we move down to a population level, we can start to think about what is the abundance, distribution, and fitness of the same group of viruses, right? And so also trying to identify what are those optimal growth conditions or replication conditions. And then finally, thinking about evolutionary ecology. So this would be perhaps the study of viruses that considers the evolutionary histories of a group. And in this case, what we're hoping to do, which we're still very much in the process of, is what fitness adaptations have occurred through infection cycles. So now we're trying to drill very far down, not only in terms of looking even subpopulation at the cellular level, but those individual cycles of infection, what happens and how does that maybe change and lead to sort of the development of some of these bigger phenomena that we see in the environment. So try to move this one here. Okay, so this is work that we are doing off the coast of California. So this is sort of at that top level, thinking about viruses and community ecology. There's also work done by a PhD student that I'm working with. And so she has gone out on what's called the California Current Ecosystem Long-Term Ecological Research Project, or CCELTER, also a mouthful. So basically what this project is doing is looking at upwelling zones along the coast of mostly Southern California. So we are down here in San Diego, but also, so pretty much, I guess this would be considered mid, I don't know, California, but basically off the coast of California, looking at the California Current. And so what this project typically does is what they find using a seesaw, which is an instrumentation that they stick out in the water. It flows and it follows filaments of water, and it identifies areas of upwelling. And one way that it can do this is temperature, right? So upwelling occurs along the coast where you have winds moving, typically in this case along California, we have winds moving south, which causes the water to move offshore. And as that water moves offshore, that deep water is pulled up, right? And that deep water is cold and it usually has higher nutrients. And this can create a system of upwelling, and it can change the community, particularly in terms of the phytoplankton community that are there. And so right here, you see that there's a filament right here. So this is temperature, so red being hot, and these cooler colors being cold. And so you see this filament coming off the coast, right? So you see that cold water that's being brought up near the coast and moving offshore. And so in this kind of system, they call them cycles, where they go out and sample, and they cycled they sampled, my children are trying to come in. They cycled 4 different cycles within this upwelling filament. And so again, this is temperature shown here, and you can see the blue being cold and the red being those warmer temperatures. And so what she wanted to do was get a sense of not only within the filament, so what is going on with active upwelling, but what's also happening as you transition outside of that. So really thinking about the community ecology, again, in space and time. And to study viruses, she wanted to use what's called a modified dilution assay. So in this assay, oftentimes researchers are looking at grazers and the impact grazers have on phytoplankton mortality. And there's also what's called the modified dilution assay, which is looking at viruses. And so very basically what you do is when you're out there, you take whole seawater and you filter it, and you get different sizes, right, of size classes of organisms, and then you start to dilute out the whole seawater. And as you're diluting out the whole seawater, right, you can think of that you're diluting out, say if you start at 100% and you move to 20%, you're diluting out the grazers or viruses and pretty much the whole community, right, in those levels. And then you come back to the lab and you measure and evaluate who's present and also what is the phytoplankton growth rate, right? So here's the apparent growth rate. And this is just a stylized, it never looks this good, but what it shows you is that, so if this is, say, the control, well, I shouldn't say that. In our case, it's the control. So if this is the 0.45, so this is often what's used in grazer assays, right? So they have taken out the grazers, but the viruses are still there. And so if you compare, if you take out the viruses, right? So now we're looking at 30 kilodalton or smaller. So this is really, so basically ultrafiltrate. So there's nothing in that water, no organisms, no viruses. If we look at that water and we compare it, we can say, well, is the apparent growth rate of the phytoplankton in the system changing where this line is steeper? So it's more negative. That means that viruses are directly impacting those phytoplankton. If it's below that, typically what we would say is there's an indirect effect to the phytoplankton growth rate. So that's a very basic way of how we're looking at these types of experimentations. And so when she went out to these different cycles, right, that are in these different filaments, we of course see very different trajectories. And so there's a lot going on here, but so again, this is the dilution factor rate. So you start at 0 and you go to 1. This is whole seawater. This is all 100% seawater, and apparent growth rate on the y-axis. And just what I really want to show you is that, so again, we have the same kind of lines as I mentioned. They're not as pretty in real data. So you have in solid the grazer free, and then in the dash the virus and grazer free. But really what I wanted to show you is just this line. So what you see here is this is the slope, okay? And so what you can see is that in cycles 1 and 2, where you have the upwelling water, we definitely are seeing a stronger, more direct impact of viruses on the phytoplankton, as opposed to cycle 3 and 4. And I don't want to go into it too much, but what's coming out of this study has been really interesting, because one thing that Sarah sought to do was something that most people don't like. So often what happens is that you count using microscopy, which is flawed in many ways for viruses, because you only see mostly bacteriophage, actually, which is not really what we're going for in this kind of study. We're really hoping to see some of the phytoplankton viruses. You also don't know who you're looking at, right? So when you're counting under the microscope, you just see dots. And so you don't know what that virus is. And the same thing for phytoplankton. What they do is you typically count using flow cytometry, different size classes, which is great, but they're very bulky, right? So they'll be above 20 microns maybe and in between and then down. And so what she wanted to do is she sequenced everything. She sequenced the whole community, 18S, 16S, so conserved genes. She also sequenced their RNA to look at activity, and she also sequenced the viral component. And now she has pieced it all back together, which is very surprising. But what we're finding is that these assays are really showing us there's a lot going on in those groups. So those large groups that are often looked at that are glossed over. So there are entire changes between the phytoplankton that are occurring that you would never see if you just looked at those large size classes instead of actually sequencing the different components. And so another component that she saw is in cycle 3 and 4, it looks like viruses are playing no role, right? So if you look at that slope of the line, it looks like there's no direct impact of viruses, but that's actually not the case. And what we think is happening is that in these systems, right, you have very warm water comparatively, right? So this is way out here. It's not in this nice super cold water that's near the shore. And it's also nutrient deplete by this point, right? Because it's moved offshore. Phytoplankton have mostly eaten all that material up shore. And so another caveat to this kind of assay is that what's done is that you add nutrients to the bottles before you incubate for 24 hours. So you add nutrients, you incubate for 24 hours, and then you sample. And what you're doing, right, is you're selecting for whatever populations might still be in that community that are going to chew up those nutrients. And so you're actually artificially making an experiment that you probably didn't want to do. And the rationale behind adding the nutrients is important, is that when you put your communities in a bottle, you don't want them to die, right, in the 24 hours. So you want to make sure that they're not limited in any way and that you see those interactions. But unfortunately, what is occurring, what Sarah has found, is that you are perturbing the system artificially. And so she found this in part by new data where she went back out. So 2 years later, she went back out on the same kind of research cruise. And in this time, she took different samples, but I just want to back up to that first cycle. I forgot I had put this in here, but so one thing that we found, which was pretty interesting, is that we've been able to link different viruses to their hosts. This is an example of just one, which is Cathosaurus, which is a diatom. So diatoms love to be particularly near shore, and they like to be in these upwelling environments. They're very good at outcompeting other phytoplankton for nutrients. And so what she was able to do is show not only that there is Cathosaurus, but there's also a link between the virus itself. So right as virus goes up here, we see a reduction of Cathosaurus in those samples. That all kind of makes sense. One other thing, though, that was pretty interesting is that we've been able to track offshore transport of active infections, particularly of some of these diatom communities. But then again, when looking at her data and looking at these mortality rates, as well as some of the specific characteristics between host and virus, it became very obvious that we were seeing some trends that we couldn't really make sense of. And so what she was thinking again is that maybe what's going on is there is this resource competition that we are developing within the bottle just because of inherent to the assay that we're running. And so what she wanted to do was in this secondary one to run an experiment where she added no nutrients and she added 10% of the normal nutrients that are added in a typical experiment, as well as repeat the experiment that was done originally. And then the other component that is often overlooked in these assays is they only run for 24 hours. Well, for pretty much all phytoplankton, eukaryotic phytoplankton that I know, that would be not long enough to see a viral infection, or not long enough to truly track a viral infection, because they typically need at least 48 hours, if not longer. And so she tested some things in the lab to make sure that everything was not going to die immediately. And then she ran her experiment for both 24 and 48 hours to evaluate whether or not we were in what's called the Eclipse Period. And so that is shown here. So the Eclipse Period refers to when the viruses have attached or been absorbed into the cell, but right, they need time to replicate and time to repackage themselves. And so that Eclipse Period is when that's occurring within the cell. And so another study we wanted to do was also looking at virus dynamics through time. And so in this one, we used what's called the ESP, or Environmental Sample Processor. And I think I saw that Chris Scholin is going to talk, or someone from his group, who is amazing. And I absolutely love to use the technology that they develop. And I've been fortunate enough to use the big guy shown here, as well as some of their new, I think that it's still 3G, it might be a 4G now, I'm not sure, but which is much smaller and on AUVs. But these samplers have really been able to sort of explode our understanding of marine microbiology, I think, because it allows us to sample in space and time on relative timescales to biology, right? And so this is particularly true for viruses because, as I just said, they're infecting, 48 hours for phytoplankton, much less for bacteria. So if we're really going to track what's going on with these different infections, I need to hurry up, track what's going on with these different infections, we need to be able to sample throughout time. And so this is one that came out last year, but looking at viruses over about a week in time. And what was found was that we see different diel activity with viruses, where we see night expression of, particularly looking at phytoplankton viruses, of both Astrococcus and cyanobacteria viruses. And in the day we see, or we at least recover, more single-stranded RNA viruses. And I just put this caveat in here, just remind everybody that when we're looking at RNA viruses, we never know if we're looking at their genomes or their expression yet. We're working on that, but that's going to take some more method development. So the other component that we noticed within this study, as well as other studies looking through time, is that there seems to be a difference just very generally between these viruses. So remember, now I'm grouping them by their genome, and I had said that there was these different genomes. So double-stranded DNA viruses are what the majority of viruses that we know about are. They're also the majority of phytoplankton viruses. So most eukaryotic phytoplankton viruses that we've studied in the past, or that others have studied in the past, have been these large, typically double-stranded DNA viruses. But what we are interested in is more of these RNA viruses, and RNA viruses infect diatoms and dinoflagellates we know of, and a few other groups. And what we see is we just see different either expression or different sequence recovery patterns between these very broad groups, in that we see a coordinated expression between double-stranded DNA viruses and their host, and we see a lag for the single-stranded RNA. And the thinking here is that it's possible that because those viral genomes are so large, they need to be coordinated with their host, right? So that they actually can replicate their genomes, because that's going to take a lot more energy and a lot more time than a small single-stranded RNA virus. And the difference in size can be, most of the RNA viruses that I study are about 8 kb, 8.5 kb. And the phytoplankton viruses of the group is called Phycotiniviridae, that infects other eukaryotic phytoplankton, these double-stranded DNA, they can be 500 kb, right? So there are huge differences in size between the two. And so one thing that we suggest, so two, is looking at these two very short time periods, day-night periods, suggests that there's a subset of the population that's infected and lysed multiple times just during this small time course. And to hurry, I'm actually not going to show this, even though this is like my favorite. But this is where we went back using the ESP, and we just sampled, over a longer time period. And what we were able to find was that there was a viral role in ecological succession. So basically, well, I'll go through real quick and then I'll just jump through some other stuff. But so this again was where we looked through 1 month, and we were able to take samples oftentimes multiple times during the day. So time is on the x-axis, and the group total or relative sequence abundance. So we're using this as a proxy of presence, right, of the organism, is on the y-axis. And so I've just pulled out some of our main groups. So in green is diatoms. In gray are dinoflagellates. In yellow is nitrate, which is shown on this axis here, this other y-axis. And then in red is the diatom viruses. And so these samples were taken off the coast of Monterey, and it has long been shown that there is this dynamic between diatoms and dinoflagellates, particularly in this area where you see this ecological succession. It's very normal, natural, and it's actually very good for the system. But it is always, to my knowledge, mostly been sort of tabled as either light or nutrients that are causing this succession. And so what I wanted to see is there must be some viral role out there, possibly. And if so, what is it? And so this kind of leads, this is sort of an oversimplification of the differences between dinoflagellates and diatoms shown here. And I'm not sure if you guys have already spoken about this, but if you put nutrients or light here, typically diatoms and dinoflagellates are kind of at opposing ends, right? So diatoms, and this is of course oversimplified, but diatoms, we think of them near upwelling regions where it's very turbulent and high nutrients, and then dinoflagellates would be on the opposite spectrum. And so basically what we saw when we looked at this was that we see this spike of nitrate and immediately the diatoms respond. This is very well known. They, as I mentioned, they outcompete other phytoplankton for nutrient uptake, but they also can store nutrients, which is pretty cool. So they can usually not only outcompete, but persist and kind of take over the system and bloom. And then what happens is that, interestingly, what we started seeing was this spike, again, lagged of the diatom virus, right? So it comes up, we see the coordinated reduction of diatoms, which you would kind of expect. But well, this part is what was the most interesting, is that now we have another secondary nitrate peak, larger than the first, but this time the majority of the diatom population can no longer respond, right? Because they've been taken out by the virus. And now we see the dinoflagellates able to respond and be in this environment and take over the community, or I shouldn't say take over, but sort of come up in the community. And so just kind of reverting back to this, these studies are really kind of how we're putting viruses into this sort of ecological framework, looking at, again, how they change through space and time, right? So looking primarily off the coast of California, and then also trying to look at what are their optimal growth conditions, or where do we see them grow optimally, right? We're seeing these differences between double-stranded viruses and RNA viruses of the major phytoplankton groups that we're looking at. We also see that there is, just like their hosts, there's a difference in distribution of the viruses. And so I can stop here and just not talk about the single cell work, or I could maybe go quickly over the single cell work. What do you think?
I had to find my unmute. Maybe you should show a little bit of your single cell work, because I think that's really neat, very new.
Okay. So
and challenging.
Yeah. Yes, it has been, but it's been really fun to develop some of these methods and to either or modify existing developments, and so or methods. And so basically what we've done is mostly talked about community and population, but really looking at the population, particularly for phytoplankton, is problematic when thinking about viral infection, is because we know that not all of the population, right, is infected. And we just saw that in that nice plot. You can see that there's clearly a subset of diatoms that were resistant to viral infection because they were able to come up with that nutrient spike of nitrate spike. And so what we've been trying to do, again, is develop these emerging model systems, and particularly in diatoms, because I love RNA viruses. And so for diatoms, so far, only small single-stranded DNA and single-stranded RNA viruses have been found, which is very different compared to the rest of eukaryotic phytoplankton. Well, not the rest, but the majority of eukaryotic phytoplankton that we know about. And so what we decided to do was to dig a little deeper into RNA viruses. And the reason being that I think partly that they're so exciting is that they lead to this vast genetic diversity within a single round of infection. And this is due in part just to their biology, right? So they have high rates of mutation. So here is mutation rate plotted here, and here's genome size here. So we would be down here. And then you have RNA viruses way up here. So their genome size is very small and they mutate very rapidly. This is a way for them to adapt, right? To continue to infect and adapt on a time scale that is generally more rapid than their host. And they also can produce large population sizes. And this just shows you here, this is a model where all of these different little ticks and circles and triangles and everything show you within one cell, this is what would happen within one cell, the difference, the genetic difference of each virus. And so 27 viruses plotted here. So you can just see that there's just a lot of genetic variation that is made through a single round of infection. And so we picked my favorite diatom, because I think it's the prettiest. And it's the most fun to look at under the scope because it just forms these really long, it's a pinnate diatom, and it has these little I don't know, at its end, and you probably know much more about them than I do, but they're just so wonderful to look at under the scope. They have these like kind of little bowls at the end, and they love to just glide and move all over underneath the microscope. So they're really incredibly fun to look at. And we can also isolate them everywhere. We also found them, which is unpublished, but hopefully coming at some point. We've looked all over the globe at RNA viruses that infect phytoplankton, particularly diatoms, and we find slender theca pretty much everywhere we look, and their viruses. And so we set out to isolate them. And so what we did was culture them. This is just shown here. I'll just to move faster. So this is what a culture looks like, where you have a control, no virus added, and this is treatment. Clearly they're infecting, clearly they're unhappy, right? You can see. And here's the virus here. So this is nice electron microscopy of the virus. And this is a not so happy cell also, which appears to be full of viral particles. And so we were able to isolate the virus. We also developed some methods that we're now using on our environmental data sets, where we can actually pull RNA viral genomes away from the rest of the transcriptome, which is kind of fun. But we were able to place it within, this is the diatom virus group. So that just makes us feel good. We have this nice virus, we have its whole genome. So now we can go and start to use some new technologies, right, to look at single cells, because now we'll know what we're looking for. In addition, we had to sequence the host genome. And this became apparent right away when we started doing single cell work, because we were mapping to transcriptomes and we realized that does not work at all. And I thought it would, but it does not. And so we had to go back. We sequenced the slender theca genome, which has actually proved to be incredibly fun because it's very different than all the genomes that the other diatom genomes that I've looked at. So we've been looking at single cell technologies, and we've been using 4 different single cell technologies. We use micromanipulation, where you can actually pick a single cell. We use flow cytometry, a process called dropseq, which I'll show. And then we're also just starting to use capillary flow. And so this is the picture, right? So we've been able to, you take an image of your cell as you're flowing it and as you're picking a single cell and putting it into the well. So you know not only what the cell is, but you also have a nice image of who it is. So this is dropseq. So dropseq is a microfluidic way to isolate single cells. So this is what the chip looks like, and each of these could be an experiment if you wanted to. So there is a couple different components here. And so this was developed for human cells, and so we had to manipulate it slightly to make it work for diatoms, and also test all of it for diatoms. But so you want your oil inlet, which is shown up here. Then you have your cells, right, that you add. And then you have your beads. And these beads have little barcodes attached to them. And so on those barcodes, we're able to use those to map back to every single cell. Okay. And so this is what it looks like. So you're looking underneath the microscope and watching this happen. So we get to watch this occur. And so this is where they're all coming in at the very end. So this is right here, where you're actually forming the droplets. And so this is what they look like when they come out, right here. So, and if we examine this a little further, here is one droplet with a single cell of slender theca, and then here is the bead, okay? And so what happens next is that we lyse the cell within the bead, and then all of that RNA is captured by those barcodes on the beads that we can wash off, and then we can sequence. And so this is what an RNA profile looks like, the messenger RNA, or the transcript profile of a single cell of slender theca. So, and there was a lot of testing that went into each different stage, which obviously is not fun to talk about, but is an incredible amount of work, but it does work. And so what are some of our initial findings? So these plots here are showing you richness, so the number of genes that we're hitting to the genome on the y-axis, and then the depth, so the number of reads that hit there. And so some of just our very, very initial findings is that these are different cell counts. So we've tried to sequence multiple cells down to single cells and evaluate them. And what we see is that we see differential expression patterns, right? And you can really see this within, when you're looking at 100 cells, there's clearly a different number of genes, even though the depth is staying the same, right? So there's definitely some kind of population level heterogeneity within the system, right? And then we also see, which I I haven't seen. I keep looking for someone so that I can feel better about this data, publish this. But what we see is that for our single cell data, there's between 1,000 and 2,000 transcripts per cell for the diatoms. And this is for Slender-Theca, as well as we've tried to look at a few other model diatoms. And it seems to be pretty standard in that this would be sort of the baseline number of transcripts per cell that we're finding. Which these are very basic numbers, but they become quite powerful when you're looking at modeling the system. And then just very generally here, if we take another look, looking at bulk, when I say bulk RNA-seq, that's the typical RNA-seq where you're doing the whole population, the whole culture at once. It would be here in green. And then we have test single cells using both drop-seq as well as flow cytometry. And then we have viral infected single cells here. And what you can see is that as well as bulk viral, I'm not showing it here, but anyways, what you see is that the viral transcriptome, as you would assume, of the cell is depleted, right? Compared to, say, a healthy one over here. Well, what does this all mean? So we're starting to put this all together. And so this is just a snapshot of some of the data, but we've been looking at tens of thousands of cells at once. So now, right, we've done community, we've done population, and now we're going back down to this, all the way down to the single cell level to then extrapolate back up to what's going on in the community, because we clearly see that there is heterogeneity within the system. So using whatever method we're using, and if we look at genes, all the genes of the Slender-Theca genome are shown here, and these are all different cells that we're looking at here. And the colors indicate whether or not you have high levels of that gene versus low levels of that gene, red being high, white being low. And so you can see that there definitely seems to be populations, right? This group of cells, this group of cells, and this group of cells. And when we do some bioinformatics or computational biology using, say, T-SIN plots, you can start to see those subpopulations come together. And those are shown down here for both the drop-seq data as well as the flow data, that we are getting these subpopulations and this population heterogeneity that we're now able to say something about, not only in terms of, is it infected, is it not infected, is it stressed, is it not stressed, how much of the population is stressed and infected. So those are the types of data that we're now pulling from these experiments. And we're also working on a model, which was really fun as well. So we've been able to piece back together a genome metabolic model for Slender-Theca, as well as when it's infected with this virus. And we found out that it grows in the dark. And this was something that our collaborators who are working on the model, Kirsten Zeigler, who's at UCSD, he said, hey, have you ever known diatoms to grow in the dark? And I said, yes. He was like, well, I think this one does. And so we went back in the lab and we tested it, and sure enough, it does. So it was kind of cool. And so I'll just end there. This is just putting it all back together, but really, again, kind of thinking right from the community level all the way down to the cellular level, and not only how different those are, but really what is the importance of it. I mean, clearly we're seeing that there's differences in each of those different levels of biological organization, but they all connect back to one another. And so hopefully what we'll learn is some of what are those connections and what are those feedbacks that are driving, say, what's happening on the single cell level all the way up to what we're seeing within community and food web transitions. That's it. Sorry for going over.
Okay.